Good afternoon, dear members of judging. I'm Andraj. I'm from Kiev, Ukraine. Today, I want to tell about own science project, namely in silico research of the potential role of a number of compound class N-acetylamines as inhibitors of a protein of coronavirus SARS-CoV-2. But suddenly arises a question: why it's so really important? Because now of the global pandemic of COVID-19, acutely arises a question about searching a new biological active compounds that are able to possess an anti-coronavirus properties and developing based on them safe and non-toxic drugs. However, the main problem of design of these compounds that is time for creation not enough. And thus for this problem for help come computer innovative technologies, for example as molecular docking or artificial intelligence and others. Therefore, the purpose of this research become an experiment in silico, research potential possibility of the N-stereotylamine and other N-acetylamines being with the active site of a protein of coronavirus SARS-CoV-2. But for this purpose were set the following goals. Firstly, prepare or study the spatial structures of N-acetylamines. Secondly, with method of molecular docking, assess potential possibility of interaction of the N-stereotylamine and other N-acetylamines with active site of a protein of the coronavirus SARS-CoV. And thirdly, identify the most probable compound for further in vitro research. Well, at the start, special structures of N-acetylamines were downloaded from different internet databases, for example, as Protein Data Bank, Drug Bank, or PubChem, or were constructed independently. These structures that were constructed independently, we can see in the right side upper it's an allyl ethanolamine, and down below it's N-casapentanyl ethanolamine. And after that, molecular docking of special structures of N-acetylamines with the protein was conducted in the same program, Autodock Tools 156. Then, the lowest free bond energy defined in Autodock VNAD 112, and based on affinity calculated of inhibition constant of N-acetylamines for a protein with formula. Constant inhibition equal exponent of expression delta G is affinity, namely the lowest free energy, split on gas constant R, multiplied on temperature by Kelvin T. Next, in the left was created a table with the statistical data of each interactions. By the way, we can see two key amino acid residues that play an important role in the active site of a protein. And in the right side, we see visualization of each interactions. Also, I want to stop on this slide because we can see as example N-stereotylamine that bind with the active site of a protein of the coronavirus SARS-CoV, where in mesh spheres shown electrostatic field of interactions. So, in the results of research indicate that all studied N-acetylamines can bind with active site of a protein, and in the fifth figure we can see this ability. Yeah, it's here, and we can see that all N-acetylamines that was studied in this research can bind with active site of a protein. So also in the right side, one can see comparison of affinity and inhibition constant of each interactions. In general, what we can say in the conclusions. Firstly, that all N-acetylamines are able to bind with active site of a protein, but the N-stereotylamine possesses the highest affinity and inhibition constant comparing with other N-acetylamines. That confirms the assumption of the potential possibility of using N-stereotylamine with in order to influence coronavirus SARS-CoV-2 in the organisms. And the most important, I consider that the results expand the knowledge of biochemical mechanisms of the N-acetylamines and can be used for creation a new medicinal preparation for with anti-coronavirus properties. that is the references that was used in this research. And thank you for your attention.